Great. Thank you very much. Thanks, Rhiannon, for the introduction. And thank you all for your patience while we sort out the technical difficulties. So my name is Lin Yi. I'm a fourth-year graduate student in the Zhang Lab, and it's my pleasure to be speaking with you today about the basics of using CRISPR in the lab. So I want to start off with just one photo. Who can tell me who this is? Okay, so this is Luke Cage. He's like a superhero, but most recently he's been kind of reconfigured in a Netflix series. And what's cool about him is that his superpowers were actually introduced using CRISPR. By fusing, I think, an albatross or like some kind of like animal gene into one of his genes, they make him bulletproof with some kind of like cool power. So that's really cool. It's also a very sci-fi. But the cool thing is, at least the technical part about putting the genes into him, and you know, aside from all the superpowers, the technical part with CRISPR is actually more or less feasible to do. And while making a superhero is still sci-fi, a lot of other applications, such as in disease modeling, in agriculture, and other kind of fields, are slowly becoming a reality. And the applications of genome engineering have really, in the last couple of years, sprung up and become very widespread. And today I'd like to talk about some of the general process of how we might use the latest generation of genome engineering tools to create some of these applications. Oh, and if you have questions during any part of the talk, please feel free to stop me. This is meant to be as interactive as possible. Okay, so what we'll cover in this section generally, so introduction to genome engineering and CRISPR systems, and then I'll give an overview of a prototypical CRISPR knockout experiment. What are the design challenges with that, and what does it look like to carry that out in the lab? And then I'll talk about an application that's popular for research, which is using homology-directed repair to create precise genomic edits. And I'll end with a survey of recent CRISPR technology developments. So, genome engineering. I think the heart of genome engineering lies in the insight that DNA double-stranded breaks in the genome can be leveraged to edit the DNA near where that break occurs. So this is work begun in the 80s, and largely the DNA repair falls into two different pathways once the double-stranded break has been induced. The first is called non-homologous end joining, which is basically where the cell goes into a panic mode and tries to stitch together the DNA ends however it can to prevent itself from dying. And that tends to create different kinds of insertion and deletion mutations in the DNA. And when that happens in a protein coding gene, for instance, that can result in frameshift mutations that inactivate the function of the gene. So it's very useful as a gene knockout tool. The second is homology-directed repair, which the user supplies a DNA repair template where the ends of the template are the same as the endogenous DNA, but the middle is different, and the cell will incorporate that or use that as a repair template to create precise changes. So then the question is, if double-stranded breaks are so useful for genome engineering, how do we create them in places in the genome that we want in order to edit those genes? So in the earlier years, some of the more popular techniques were based on protein-DNA interaction. These included zinc finger nucleases and talons, which basically relied on the specificity of interaction between protein and DNA to direct where the double-stranded break happens. What I'll be talking more about today is CRISPR, which actually uses RNA-DNA base pairing complementarity to determine where in the genome the editing occurs. So what is CRISPR? So this is Cas9, and it's a very large endonuclease with approximately 1,368 amino acids, and embedded within it is a piece of RNA known as the guide RNA, shown in red. And when the Cas9 guide RNA complex enters the nucleus of a cell, it goes around looking for anywhere in the genome where it matches that piece of guide RNA. So once it finds it and you have a perfect match, Cas9 will then create a double-stranded break in the target DNA. See if it's working. Yeah, in the target DNA. So this is a very sequence-specific process. When this break happens, it can either be repaired by non-homologous end joining, where you have kind of random insertion and deletion of nucleotides and scarring of the DNA. That happens predominantly. And so you have the indel mutation here, which can knock out a gene. So the other thing that can happen is if you supply a repair template, let's see this coming in here. So repair is much less efficient than NHEJ, as from the speed of the video. Then it can repair the break according to the template that's provided. Yeah.
So what's the percentage really by the non-homolog joining in the repair without error?
Oh, one more time, please.
Just a second. Since you're going to repeat. Thank you. I'm just asking, majority will be repaired by non-homolog and joining without any error in it. And do you have any sense about the percentage in regular cell repairing system?
In terms of how many times Cas9 has to cut before you get
cut, after double strand break.
Yep.
And you have two machinery. One is non-homolog joining and another one is HDR. But for the end joining, if there's indel insert, it's the error happen in the prime error. But most of time it's just by itself. It's no error, no indels caused. So what's usually the percentage goes the correct way, and what's being repaired with indels?
So with like ribonuclear protein delivery of Cas9 into a cell, just practically speaking, you can get up to 90, 95, even 100% like indel formation in the cells of interest. In terms of like the repair kinetics of whether or not the break has to happen many times for that to happen, or whether or not it's just like indel forms during the first break, I think that's still an area of active investigation. There's a couple papers on bioRxiv where they argue that usually the break only happens sort of once because Cas9 is slow enough. But there are also other papers that argue you have to cut multiple times before the break happens. But in terms of editing efficiency and just knockout, I think you can get very high levels of editing.
Are you asking also then what is the efficiency for HDR? To get a new piece of DNA in? No.
And if you have the Cas9 constantly expressed in the cells, is that theoretically you will eventually have 100% cut if it keep present the Cas9. But maximum efficiency, usually you get like 30 or 40, 50%, 60%. What happened to the rest if you still have Cas9 continuing expressed in the cells, and why they're going back? And theoretically it's because of there's a repair, don't have any errors happen, no indels. And that's what my question.
I see. So why don't you get 100% editing always? Is that the question?
Yeah.
Basically? Yeah. Yeah, so with a lot of times when you actually have constitutive expression over long periods of time, if you're delivering it via lentivirus or if you're delivering a very efficient target via ribonuclear protein, you can actually get very close to 100% editing, especially with like lentivirus-based delivery. With stuff where you're just transiently transfecting something, it's possible the Cas9 doesn't have long enough to act, or maybe you don't transfect like all the cells. It's like a transfection efficiency thing. But overall, I think unless you accumulate some kind of like substitution mutation in the target where you don't allow it to sort of cut again, most of the time, in theory, you should actually be able to get very high levels of editing. I think it's mostly a delivery thing that's causing like low levels of editing, or maybe your guide doesn't work as well. But for a very efficient guide, it should be quite good. Yeah, that's true.
So one other reason why you may not get to 100% is Cas9 may cut near the PAM, or it may make when it cuts at the target, it may create a larger indel that gets rid of the PAM. And so if you see things like that happening, that can also reduce the efficiency. But yeah, I mean, you should get close to 100% with a good guide. If you're not getting close to that, you should design a different guide.
Sorry, one more question. So if that's the case for in vivo application, if you have, like, for example, AAV in a certain cells, which is not dividing cell, and then you constantly have your Cas9 expressor for, like, say, a long time, and eventually you're expecting in vivo application, you will have 100% cut, theoretically. Is that correct?
I think for AAV delivery, it's very hard to get it into every single cell. But if you actually have it expressed at high levels in any given cell, I think you can be pretty confident that that cell will eventually accumulate and edit. But that's not the same as saying, like, you'll get 100% editing with your AAV.
That means as long as you have AAV in it, you have Cas9 expressed with guides together, you will eventually have cut in the cell, have editing.
Yeah, given long enough, that particular cell should saturate with an edit. Yeah.
Will be saturated.
Yeah. Yeah. Thanks. Okay, so CRISPR is a naturally occurring bacterial immune system that's used to defend against foreign nucleic acids that invade bacteria, but it's been repurposed as a mammalian genome engineering tool. And there are two classes of major bacterial kind of CRISPR systems. One is class 1, where you have the effector enzyme kind of divided into many different different protein complexes, and class 2, where it's a single subunit CRISPR RNA effector complex. And these are the more useful ones for genome engineering because you only have to worry about delivering like a single piece of protein. So a lot of the work in genome engineering has focused on class 2 CRISPR enzymes. The most popular one is Cas9 from S. pyogenes. So this is a ribonuclear protein complex with a guide RNA. Let's see if we can. Okay. Well, with a piece of guide RNA, and the two main things is that it recognizes any target that's complementary to a guide as long as it's adjacent to a short DNA sequence known as the protospacer adjacent motif, or a PAM. In the case of Cas9, it's a trinucleotide sequence, NGG. So as long as it's there and the guide matches, it should have a reasonable chance of cutting. So different orthologs also have different PAM sequences, which I'll talk about later. The similar thing, you can target either strand. You can just kind of flip the diagram on the previous slide over, and you can, so the both strands are symmetric. So either a target site on either strand can be targeted nicely by Cas9. So this is the crystal structure of Cas9. The most interesting features are that there are two nuclease domains, one of which is responsible for creating a break in each of the strands. And by mutating these nuclease domains to make them inactive, it's possible to create Cas9, a dead version, which just binds to DNA sequence specifically without actually cleaving it, which is also useful for many different kinds of engineering applications. As a note about the biochemistry of how Cas9 works, so it starts off by recognizing the PAM bases, and then tries to unwind the DNA starting from the PAM by using the guide RNA complementarity as a tool. As a result of this biochemistry, the DNA bases that are most proximal or close to the PAM have the least tolerance for mismatches, and the ones that are near the end of the guide, away from the PAM, have the most tolerance for mismatches, which is important to keep in mind for considering off-target effects and specificity. So now I'd like to overview a prototypical CRISPR knockout experiment and what that would look like. So the first piece of a design, let me just actually go over the timeline. So it starts off with just designing the guide sequences, the donors, and all the oligos that we'll need. And then constructing the reagents, whether that be plasmid or virus or other delivery modality. Delivering the Cas9 into the cells and functionally validating how well the Cas9 worked. And optionally at the end to do a clonal expansion to get your cell line of interest. So for in silico design, how to design guide RNAs. So there are many tools available, but a couple of important things to keep in mind are just the species that's being worked with, as well as whether or not you're doing a knockout or a knock-in. For doing knockouts, the selection of guides is much more flexible because you can knock out a gene in many places and it'll still inactivate it. Whereas for specific knock-ins, you want to get much closer to the actual site of interest. So there are many great tools available online. The one that we use most often in our lab is Benchling, which is provides a very nice integration of both the state-of-the-art on-target guide efficacy scoring, as well as off-target scoring, all in the same kind of integrated software. So here, for example, you see I just put in kind of a random gene. This is from EMX1, and selected a region within the gene and had Benchling calculate all the different guides with the on-target and off-target scores. And this is really nice. It only took maybe like 10 seconds to generate this image. And this is a very robust list of guides, and I can just go and pick the ones with the highest on-target score and a reasonable off-target score. Yeah, so there are also many other tools available. It's probably too many to go over, but there's a nice review article that summarizes a lot of these tools. And if you're interested, you can look more into this paper for other availability. So after the guides have been designed, we need to figure out how to deliver the Cas9 as well as the guide into the cell or animal of interest. And there are many delivery methods, but I think the three arguably most popular ones are either with plasmid, with a virus, either lentivirus or AAV, or by direct delivery of the ribonuclear protein complex. So for the first two, actually let me just go over generally comparing them first. So there are many great plasmids available where there's a promoter driving Cas9 and then the U6 promoter driving the expression of the single guide RNA with a host of different selection markers that can be chosen. For lentivirus, this is where you want to actually get the Cas9 gene or the guide expression gene to integrate in the genome to maintain stable expression over many cell divisions. For AAV, it's mostly for in vivo work. If you have this will be like a non-integrating version where you can get it to maintain stably inside a non-dividing cell for years at a time. And finally, direct delivery of Cas9 guide RNA complex, which avoids nucleic acids completely, or at least avoids DNA completely, and it tends to be lower toxicity that way. So for the first two, in plasmid and viruses, a lot of great constructs are available on AdGene from our lab and from many labs around the world. There's a great resource page where you can kind of find a list of all the different CRISPR-associated plasmids on AdGene. There's many orthologs. There's different kinds of, all sorts of very rich collection. Here are some general Cas9 purpose reagents that our lab has produced that tend to have been the most popular over the recent couple of years. These include the PX330 series, which is Cas9 and guide RNA with various kinds of selectable markers. Lenti-CRISPR, which is Cas9 and guide RNA inside a lentivirus plasmid. And then an AAV plasmid, which is actually a smaller Cas9 that we've characterized that fits a lot better inside an AAV backbone. It's also highly efficacious for genome editing. So for use of the PX330 series plasmids, it's basically good to go, except you just need to paste in the guide oligo sequence. So this can be ordered as a short, like 24 nucleotide sequence, two of them from IDT, annealed together and pasted in between the two BBS1 restriction sites. So this is a couple of hours of cloning, and then the guide cloning is usually very efficient, where you can get 90% of the colonies on the plate to be correctly cloned. So after constructing the plasmids, you can deliver them by transient transfection into the cells. So for like HEC293T cells, for instance, I generally put in about 500 nanograms of the plasmid per 24 well. And at the end, for a really good Cas9 guide, that can get 60 to 70% up to indel efficiency. It's also a good idea to include a GFP control to make sure that the transfection is high efficiency. We've also profiled kind of how long it takes for the transiently transfected cells to reach the maximum editing efficiency. And I think it basically kind of curves up until about 60 hours where it reaches the saturation point. And it doesn't go any higher after that. So for virus production, Julia is giving a talk in the next session, and she also has a great paper out about some protocols, so you can reference those. And finally, I think one method that might be a little bit underemphasized generally for Cas9 delivery is direct delivery of the protein complex. This is a very attractive method because it avoids DNA delivery that can be unwanted, incorporated into the host genome in unwanted ways. So there's many commercial sources of Cas9 protein available, and the guide RNA can either be produced in the lab or you can actually just buy it directly. And if you purchase the guide RNA directly, you can complex it with Cas9 and do the delivery immediately without any processing steps. This tends to avoid drawbacks such as like any toxicity associated with plasmids. And for a good guide, RNPs work really, really well. You can get like 80 to 100% editing range for a good guide RNA. Okay, so after the CRISPR reagents have been constructed and delivered, after a few days, we can functionally validate how well the Cas9 worked at that particular target site. And we can assay a pool of cells for both on and off-target editing. So there are three main ways that I've encountered where people have been able to assess Cas9 editing efficiency. The first and arguably the most popular is called a surveyor assay or T7E1 endonuclease assay. It's a gel electrophoresis-based quantification where higher cleavage activity results in greater intensity of cut bands. The second is called the tracking of indels by decomposition, or TIDE, basically based on the principle that if you Sanger sequence a mixture of different amplicons, the sequencing trace that results from the mixture can actually be decomposed in such a way that allows you to back calculate how much indel there was in the original pool. And finally, there's also using targeted deep sequencing, which I'll talk about in a bit. So after Cas9 is delivered, a collection of indels are generated. It might look something like this. We want to figure out what percentage of them are the wild type and how many have accumulated these small insertion and deletion mutations. So the way the surveyor assay works is we first amplify the locus of interest to create a pool of different oligos with both the wild type and also with the indel mutations all in the same pool. And then after they're amplified, we melt all of them and then re-anneal them. So the indels will, with some rate, get annealed to the wild-type sequence, so there will be a little bulge or a mismatch in the DNA. And then upon incubation with the surveyor nucleus or the T7E1 endonuclease, the ones with the bulge will actually get cut at a specific location where the bulge is. And generally, the bulges occur exactly where the Cas9 editing has occurred, which results in very distinctive band sizes. So how this would actually look like on a gel is this is the kind of pool of amplicons containing both the wild type and the indels. It's a single band because most of the indels are small enough where you can't resolve them just on a gel. But after annealing them and treating them with the surveyor nucleus, two cut bands corresponding to these fragments start to emerge. And then there's a formula for calculating the intensity of editing. Basically, this formula is just saying that the proportion of unedited alleles squared is the intensity of the uncleaved band in the gel. So basically, like if you think about like sampling from the collection of amplicons, the probability of getting two perfectly annealed ones of the wild-type sequence is basically the square of the relative concentration of it. So the other method is TIDE, which you basically do the same set of PCRs as surveyor, but instead of annealing and running it out in a gel, you can send it out for Sanger sequencing to get kind of a messy trace where you get a mixture of peaks. And by comparing the unedited one with the unedited one and using a sophisticated processing algorithm that's available on a website, you can back calculate not only the indels, but also the kinds of sequences that resulted in this particular sequencing trace. And then the third and arguably the most sensitive method is targeted deep sequencing, where we do a similar PCR as surveyor and TIDE, but instead we do single molecule Sanger sequencing and count for each of those single molecules. Was there an indel or was there not an indel? This is a little bit more involved of a library prep, but in terms of the pros and cons of these methods, it tends to be the most sensitive. So both surveyor and TIDE, I think it's hard to quantify below 1 or 2% indel. Surveyor also has the drawback of just underestimating indels because sometimes your bulge just isn't big enough for like the surveyor nuclease to be able to recognize and cleave it. So the intensity of the edited bands will be generally lower than the actual indel rate, especially for Cas9. For cost, next generation sequencing is the cheapest method, but it also requires you to have many different samples to justify that. So I think if you're just quantifying like indel rates for 1 or 2 samples or a small number of samples, just to assess unambiguously whether editing occurred, these two methods are probably the way to go. But to get very quantitative assessment and sensitive assessment with a large number of samples, then deep sequencing should be the method of choice. Okay, so after functionally validating the guides, we can then clonally expand them using FACS or by dilute plating, and then validate individual clones for the edit of interest. So that is the prototypical workflow of a knockout experiment. Now I would like to talk about some extensions that would be of general interest. So homology-directed repair. So previously I've only talked about using Cas9 to create kind of a random population of indels to knock out a gene. What if we want to repair it with a precise edit? So this, as I mentioned before, is a method for specifying a user specified change by providing a donor template in addition to the Cas9 complex. So some of the major considerations for HDR are whether to use a single-stranded or double-stranded donor, how long of a homology arm should be included, how far away from the cut site and the mutation in terms of the number of base pairs, and whether or not the edited allele can be recut by Cas9. So I think a lot of data that's been published suggests that if it's possible to use a single-stranded donor, it tends to be more efficient than using a long kind of plasmid-based double-stranded donor. The drawback of that is there's a length requirement for the single-stranded donor. Historically, it's been 200 nucleotides to be purchased from IDT. More recently, IDT has expanded their service to offer like really long megamers, which can go up to 2,000 nucleotides. They're also quite expensive, but maybe justified if you are looking for really high kind of efficiency knock-ins. For longer knock-ins, such as adding like a selectable marker, plasmids are a great way to do it, such as by adding a GFP. In terms of the distance to the cut site, I think for high efficiency editing, the closer the better. So here, there's a paper that profiled how the editing efficiency of HR drops off with the distance to the cut site, and it's a pretty steep drop off. So after you get to about 20 to 30 base pairs, it really declines from having it right there. So if it's possible to put it right at the cut site, that would be the best. As an interesting application of this, if your goal is to try to get a heterozygous clone, it's actually advantageous to pick it somewhere kind of in the middle here, where you don't have like super high editing efficiency, but it's right in the sweet spot where you can get a larger population of clones that are heterozygous. So how long should we make the homology arms? For SSODNs, anywhere from 30 to 90 base pairs nucleotides has been very successful. So this is a paper by Professor Jacobs-Korn's group in 2016 where they profiled different configurations of donors to see which one is the most active. And for these, having like a 36 base pair homology arm on one side and a 90 or so base pair on the other side seemed to be quite robust. There was also an effect where the symmetry of the homology arms made quite a big difference, where having an asymmetric donor where it's shorter on one side and longer on the other is more efficient than having them being the same length on both sides. So another important yeah.
You say don't linearize the plasmid.
Yeah.
Why is that?
So linearized plasmids with a linear piece of DNA, you have a much higher chance of just incorporating that into the genome in places that you don't want. To prevent that from happening or to reduce the rate of that happening, it's much better to keep it just as a circular plasmid rather than linearizing it before using it as a donor.
The homology arms can somehow figure out where they're going to go.
Yeah. So it should work fairly well with just the plasmid. So the final consideration is to prevent recutting by Cas9. So if after you introduce your mutation by HR, the same allele can be cut by Cas9 again, that will really significantly decrease the editing rate. So one way to prevent that is to build some silent mutations into the donor that destroy the original target sequence. If that's not acceptable, another really nice strategy is to do a two-step editing where you first introduce some intermediate step silent mutations that block Cas9 from recutting, isolate a clone of interest, and then you revert just those silent mutations back to the original ones while keeping the mutation of interest. This is a process known as correct, a two-step editing which can make kind of single base pair changes much more amenable to doing. So some variations of HR that can help improve efficiency or improve precision. One is using a nicking Cas9, where instead of creating a double-stranded break, you just nick one of the strands, supply the donor. And it tends to be a little bit lower efficiency for creating actual precise edits, but it's great for avoiding unwanted indel mutations. You can also suppress NHUJ repair machinery by using either chemical compounds or other suppression proteins. So by knocking down NHUJ, the rate of HR can be boosted significantly. So I'd like to survey some recent developments in CRISPR technology. So for HR, it's still quite a laborious process to create a new clone. But a very elegant strategy that's developed by Dr. David Liu here in the BroadE, as well as other groups around the world, is called base editing. So for this strategy, you can actually change a C to T directly without using HR and without creating a double-stranded break in the genome. So how this works is normally you have Cas9 create a double-stranded break, you supply some kind of a template or NHUJ, and you get a mutation there. With base editing, you actually have either a nicking Cas9 or a catalytically inactive Cas9 that's fused to a cytidine deaminase domain. And that deaminase domain can deaminate any cytidines within kind of this activity window from C to a T. And when that happens, that C to T edit becomes permanent without actually having created a double-stranded break in the process. This can be very efficient, I think up to 75, 80% efficiency in hex cells. And a great way to create this C to T mutation. A couple of design considerations with base editing is that the particular base of interest needs to fall in a particular 5 base pair or so window of the guide away from the PAM. And whether or not that guide exists needs to be computationally determined beforehand prior to using this strategy. There are other many alternatives to using SP-Cas9 for genome editing. These include other Cas9 orthologs, as well as non-Cas9 enzymes from CRISPR. As one example, our group has previously characterized an ortholog of Cas9 known as Staph aureus Cas9. It is about is over 300 amino acids smaller than the commonly used SP-Cas9, which means it can be much more easily packaged into AAV. And more recently, we've also characterized another enzyme called CPF1. So this is unusual because it actually allows you to create a staggered cut with a totally different PAM sequence. So the commonly used Pyogenes Cas9 recognizes NGG PAM and makes a blunt cut in the target. SA-Cas9 is smaller. It has a slightly longer PAM sequence that's different and also creates a blunt cut. And for CPF1, the PAM is actually T-rich instead of G-rich, and it creates a staggered cut in the DNA. One advantage of using CPF1 is that it has a very short guide RNA, which can be more easily synthesized. It also facilitates multiplex editing. So if you deliver an array of these short CRISPR RNAs with CPF1, you can edit many genes at the same time with a single promoter driving the expression of all of those guides. It's also interesting to note that the indel profile of these enzymes is quite different. So CPF1 generally gives you much larger deletions, and Cas9 either small insertions or small deletions. Another quirk is that for getting gene knockout rates for frameshift, Cas9 gives you a higher rate of frameshift mutations in CPF1 because of the presence of +1 kind of base pair insertion that's highly stereotyped. There are also many other Cas proteins that have been discovered. There's Cas12 and Cas13, which Omar and Jonathan will tell you about in the next hour. And a lot of these are still in active development. In addition to naturally occurring Cas9 and other Cas enzyme variants, there's also engineered variants that have been repurposed and equipped with different kinds of functionality. So 2 of these enzymes are called ESP-Cas9 and SP-Cas9-HF1, which have been engineered to be more highly specific than wild-type Cas9. One of these by our group, and HF1 by Dr. Keith Jung's group at MGH. So these are useful because it allows you to improve the specificity of genome engineering without making drastic changes to the guide design or the other kinds of experimental protocols. By simply substituting out wild-type Cas9 for one of these variants, off-target effects can be greatly reduced. So there are other two alternatives to reducing off-target effects as well. Either using like a paired nickase, where you effectively extend the length of the target sequence from 20 to 40 bases, or using dead Cas9 fused to two non-specific FOC1 nuclease domains. You also get the kind of 40 base pair guide extension. Both of these strategies require delivery of two guides, whereas using one of the high-fidelity variants can reduce the requirement down to a single guide. So there are also nucleases that have altered PAM requirements. So if the target site that you want to edit doesn't have the NGG, you can consider using one of these other variants. So there are SP-Cas9 variants that can recognize PAMs like NGA and NGCG and like RRT, which was published by Dr. Zheng's group a couple of years ago. And more recently, our group has also engineered CPF1 to be able to recognize PAMs other than TTT, for example, with an A or a C somewhere in the PAM as well. So these can increase the versatility of editing. If you want to characterize off-target effects on a genome-wide level, two excellent ways to do that, published earlier this year, are called CircleSeq and CiteSeq. So these are in vitro assays where you take kind of purified genomic DNA, modified in various forms, and incubate it with the Cas9 guide complex. And when Cas9 makes a cut, it creates a double-stranded break where if you sequence on each side of that double-stranded break, you can determine where the break occurred in the genome. So this is a very, very sensitive way to profile all the double-stranded breaks at the same time that are occurring in the genome, in case it's for a therapeutic purpose where you want a highly specific guide sequence. So finally, Cas9 can also be used as a screening approach, where instead of delivering one or two guides, we can simultaneously deliver a whole collection of 20,000 to 100,000 guides to knock out or activate genes on a genome-wide scale. So Julia will be talking about that in the next hour. So I want to save some time for some questions, so I will end my slides there. But I want to thank all the people in the Zhang Lab for all the support and advice. And the major resources I wanted to point your attention to are AdGene and Benchling. AdGene for plasmids and viral vectors. Benchling is a really nice platform, and you can also use it to design and select guides. We have a CRISPR Google Groups forum, which is a forum where anyone in the community can post a question or advice to receive feedback from other members of the community, as well as genomeengineering.org, which is a collection of protocols and other tips that might be helpful for designing and carrying out CRISPR experiments. So thank you very much, and I'd be happy to take any questions.
Okay. Could you comment on using RNP delivery versus plasmid or lentivirus delivery? I work in protein delivery, so I tend to hear the overemphasized, like, we should all be doing RNP complex. But I guess, why doesn't everyone do that if that's the case? And yeah, just any comments you have on that would be helpful.
Mm-hmm. Yep. Let me just put that slide. Okay. Yeah, so I think one of the reasons that the plasmid delivery, at least in our experience, has been relatively popular is that with these vectors, such as PX330 and other vectors, it's fairly straightforward to clone a guide into them. And it's fairly inexpensive to do that with a couple of dollars order of a short oligonucleotide from IDT and like a one-day cloning. The vector can just be ready to be delivered immediately. So yeah, I guess with RNP delivery, either you would need to in vitro transcribe a guide RNA from a very long template, which is a little bit more expensive, or it can be directly ordered as a synthetic guide from the company, which is also a little bit more expensive. So maybe it's in terms of like kind of just cost and timeline, where for generating a cell line where it may not issues like toxicity may not be as important as like in vivo settings or like therapeutic settings. It might just be like a little bit more kind of cost effective and time effective to do the plasmid-based delivery. But I think, I think IDT and also other companies have published some really nice data that as RNP, Cas9 can be really, really efficient. So for like knocking out one gene or for doing a number of genes, I think being able to deliver it that way actually is a really attractive strategy. I think it could be used more often. Also, RNP is great for avoiding off-target effects as much as possible. So even like with off-target effects, just by transiently delivering protein and having that only be there for a short amount of time, like off-target effects can be very significantly reduced. Yeah.
So thanks for your talk, very helpful. So there are a number of papers that actually used Cas9 with 2 guide RNAs as an attempt to sort of like delete certain sequences in promoter regions, whereby indels will not affect the promoter function. What is your lab and your experience on using Cas9 with 2 guide RNAs? I know there's an increase in off-target effects, but sometimes that may be necessary,
Yeah, so with 2 guide RNAs, when 2 cuts are made within a few hundred base pairs of each other, a really nice thing happens where a lot of the times that segment in the middle just kind of gets deleted, and you get a fairly precise joining of the DNA on either side, either just a direct joining or with like a 1 base pair kind of inserted in the middle. So that can be really good for like actually deleting sizable chunks of DNA from the genome. In terms of off-target effects, I think if you either like deliver by RNP or apply like high-fidelity Cas enzymes, or you just pick a guide that has a very few predicted off-targets, I think using 2 guides is not really significantly worse than using 1. It's just that you just have to design the 2 guides and co-deliver them into the same cell. Yeah.
So speaking about the off-target effects, do you think using 2 guides is going to cause any off-target effects? And if you look at the programs, for example, off-pointer, that will give you score values. So how realistic are those score values?
How good is off-target scoring?
Yeah.
So I think the off-target scoring is based on a lot of experimental data. At least for example, the CRISPR MIT off-target scoring is based on an experimental evaluation of how well Cas9 tolerates mismatches along the 20 different positions in the guide. So I think it's valuable as a predictive tool, but it's of course not going to be as good as you actually experimentally like assaying those off-targets in the lab. I think probably like the single biggest feature for predicting whether or not it'll cut is the number of mismatches. So if you have like a lot of mismatches to the guide, then chances of getting an off-target there are going to be much lower. So by computationally predicting a guide that just has very few of those with a small number of mismatches, it can be I think it can be quite a good way to screen for efficacious guides. Does that answer the question?
Yeah, I was just curious, like whether the deep sequencing or WG is always going to be the answer when you're concerned about in vivo work when it comes to off-target.
Yeah, so I think like for like therapeutic applications where it's like paramount to have like highly specific guide, then using these strategies of deep sequencing or like site-seq or circle-seq, I think would be quite informative. Yeah.
Hi, thanks for your talk. Could you comment a little bit more on the efficiency of using base editing without having to cleavage, and compared to the efficiency of using CRISPR with a donor?
Compared to the efficiency of what?
Of the normal CRISPR
HR.
with a donor.
Oh, yeah, with a donor. So the efficiency of base editing compared to the efficiency of HR with a donor. Dr. David Liu from the BroadE has an excellent series of publications profiling the efficacy of base editing. So I think for a really good guide, they can get about 75% of the alleles, at least for hex cells, edited from C to T. With HR, the rate is much, much lower because of concerns with like if you just have your kind of C there and it's not being, so that if the target site is not being destroyed and the Cas9 can recut the guide, then, or recut the donor edited sequence, then you actually destroy kind of your intended mutation. The other concern is just the efficiency of HR is I think lower and predominated by NHEJ. So just overall, it might be lower. So I think if you can find a guide that allows you to do base editing and the C to T change is actually what you want, then that's definitely the preferred way to go. But there's also constraints on that in terms of like being able to pick the guide that's best. Yeah.
Thank you for your great talk. Sometimes you have this cell line that is very hard to do clone. So if the clonal efficiency is very low, what would be the best way to address this problem?
If the HR efficiency is very low? Yeah, so I think either, so one strategy is just screening a lot of clones until you kind of pick one. I know a group on our that we've worked with ended up having to screen, I think, hundreds to thousands of clones to get a correct one. There is a strategy out published earlier this year where you can rely on the correlation between, like, so if one cell is getting edited kind of at one locus, then at an unrelated locus in the genome, it will also be edited. So basically what they did was you can put in not only your guide and donor of interest, but also simultaneously you put in another guide that, if it works in the cell, causes the cell to be resistant to this drug. And by leveraging the correlation between those events, by treating the population of edited cells with that drug, you actually highly enrich for the cells that have edits at both loci. And the guide that causes the cell to be drug resistant is, I mean, that genetic change is fairly innocuous, so it doesn't really impact the cell, but you can actually enrich for the correlated events like that. Yeah, I can send you the paper. It's called marker-free co-selection. Yeah.
Okay, any other general questions for Linyi before we move on to talking specifically about screening? No? Okay, great. Thank you so much, Linyi. All right. So our next speaker is Julia. She's another graduate student in the Zheng Lab, and she is our in-house expert on using CRISPR for large-scale screens. She recently published a Nature Protocols paper, which she will probably refer to a lot, but that will be a great resource if you guys are trying to get large-scale screening using CRISPR up in your hands. And she will go over the basics of both. Are you going to do both knockout and gene activation screens?
Mostly just the basics.
Okay. All right. So just kind of the basics of setting them up. And both a lot of the rules that she'll talk about in terms of setting up the screens apply to both the knockout and the gene activation screens. Okay.
Hi, thank you for the introduction. So today I will be focusing on genome-scale CRISPR-Cas9 screening. As Ryan mentioned, because this was recently published in the Nature Protocols by our lab, I will be going over the general theory of how to perform and set up a CRISPR screen. So forward genetic screens are very powerful tools for mapping specific genetic perturbations to a phenotype of interest. Generally, this involves perturbing many genes at once, selecting for the phenotype of interest, and then mapping the phenotype back to the genotype and figuring out which gene actually produced the phenotype. Traditionally, this was done by chemically mutagenizing flies, selecting the fly with a very interesting phenotype, like in this case, the red one, and then going back and trying to figure out which gene caused the phenotype. However, this was very difficult because it's difficult to figure out which gene out of the entire genome of the fly led to the phenotype of interest. But more recently, the development of shRNA screening has made this process a lot easier. shRNAs rely on the endogenous RNAi machinery to knock down base pair complementary mRNAs. So in a large pooled screening fashion, shRNAs, so the gene that was targeted originally can be readily identified by sequencing the shRNA. And since it targets base pair complementary mRNA, you can go back and map which shRNA and which gene led to your phenotype of interest. However, although shRNAs have contributed to the understanding of many biological processes, shRNAs have been hampered by lack of specificity and lack of efficiency in terms of mRNA knockdown. Now, as you know, more recently in our lab, we have engineered the CRISPR-Cas9 system for efficient gene knockout. Just to recap what Lingy talked about earlier, the Cas9 system can knock out genes very efficiently through the NHEJ pathway. The NHEJ pathway is error-prone and can generate indels, which can then lead to a premature stop codon and knockdown of the protein of interest. So the Cas9 system has so far been demonstrated to be very helpful for genome-scale screening because it is very efficient, and most guides that you design will have a fairly efficient knockdown of the target gene. And compared to shRNAs, it has generally lower off-target sequences. Besides gene knockdown, Cas9 has also been used for gene activation. The way this works is that you can make Cas9 catalytically inactive with a single 2-point mutations, and this becomes catalytically inactive dead Cas9. Dead Cas9 can now be used as a programmable DNA binder. So when we append a activation domain to it, such as VP64, the Cas9 becomes a programmable DNA activator. In our case, the Cas9 activation construct that we developed uses engineered MS2 hairpins in the guide RNA that then recruit 2 additional different activation domains to the dead Cas9 complex, and this creates a synergistic activation mediator, or SAM. And if we target this to the transcriptional start sites of target genes, then we can get very robust transcript upregulation. And so the dead Cas9 activation system can also be used for genome-scale screening. And because the Cas9 knockout and activation have proven to be fairly robust, so far both systems have been used in a number of different screens, including drug resistance in cancer cell lines, gene essentiality, viral host factors, metastasis, and more recently in the non-coding genome, as well as for cancer immunotherapy. And in theory, one can use a CRISPR-Cas9 system to screen for any biological process with what we call a screenable phenotype. So what exactly is a screenable phenotype? For screens, there's basically 3 different types of screens. So in this case, if the cells with a blue perturbation leads to the screening phenotype, then in a positive selection screen, at the end of the screen, the cells with a blue perturbation have a growth advantage. So at the end of the screen, they will proliferate, and there will be more cells with a blue perturbation than the rest of the perturbations. In a negative selection screen, this is the opposite effect. The cells with the blue perturbations will die off, and at the end of the screen, you will see a depletion of the cells with a blue perturbation. And then lastly is the marker gene selection. The marker gene selection screen is the most versatile, but also most work-intensive screen. This generally requires having a marker gene in your cells that turns the cells, for example, green. And at the end of the screen, you can sort the cells based on the marker gene expression and select for the highest and lowest expressing cells to determine which perturbations gave rise to this particular perturbation. So now I will go into more detail on the workflow of a genome-scale screen. So starting with library construction here. For library construction in the protocol, we provide for several different types of library construction. First, there are many ready-made libraries on AdGene. These are libraries, for example, for knockout that have been published. The knockout library is provided in a 1-vector or 2-vector system, targeting either the human or mouse genomes. And these generally provide 6 guides targeting the 5' constitutive exons. For the activation screening, we have a 2-vector or 3-vector system. And these screens also, again, target the human or mouse genomes, and they have 3 guides targeting the promoter of all annotated RefSeq genes. And we also provide for custom targeted library generation. For the targeted libraries are when we isolate a subset of the sgRNAs from a larger pool of sgRNAs. And this is most useful when we are only concerned with a subset of the genes, or when we want to reduce the number of cells required for screening. Because as I will mention later, the number of cells required for a genome-scale screen is typically in the order of hundreds of millions of cells. And sometimes when you're screening for primary cells or very sensitive cell types, you may not want to expand to a genome-scale screen. So we also provide scripts for isolating guides from the genome-scale libraries to construct a targeted library for these situations. And so for the general custom library design, we have a script that is very versatile. You feed it a genomic region of interest, and what the script does is it identifies all the guides in that region, and then selects guides based on minimizing the off-target activity and potentially maximizing the on-target activity, and then filters according to other criteria that may hinder oligosynthesis. And generally for these custom library designs, we recommend including multiple guides per gene. And because if one guide leads to an off-target that leads to a screening phenotype, when you have multiple guides targeting the same gene, and then you could require that all of the guides are enriched in your screen for you to call that gene as a positive hit, then this reduces potential candidates that are called because of an off-target of one guide. And we also recommend including non-targeting guides on the screen. The non-targeting guides help you determine how likely it is that you have like a false positive, for example, and also how much noise you have in your screen. And it's very helpful when you're setting up a new screen that hasn't been done before and for debugging. So for custom library designs, we have a fairly robust custom library cloning protocol. This starts with ordering the oligos from companies such as Twist or Custom Array, PCR amplifying the oligos, and then restriction digesting the plasmid, then Gibsoning the PCR amplified oligos into the backbone. Then we also provide a protocol for amplifying this library. And the important thing to remember during the library construction process is that, like all the rest of the screen, we need to maintain representation. So for example, we recommend scaling up the PCR reactions to 10 reactions instead of just 1 for genome-scale libraries. And we have a table that says how many reactions you should do for each step for example, a library of 100,000 guides. Right? And at the end of the library construction process, the first thing we do is to do an NGS verification of the library. This is very, very important because screening is actually a lot of work, it's a lot of tissue culture. And so we want to make sure that the library is actually good before starting out, especially with custom library cloning. Sometimes something goes wrong in the cloning process and your library isn't actually as good as you think it is. And so it's very important to do this step and make sure that the library is good before proceeding onto the screen. So then the next step is the lentivirus production. For the screen, we recommend lentiviral delivery because lentivirus integrates into the genome. So for any positive or negative selection screens, this is very helpful since the lentiviral genome will amplify with the cells. So then your guides will become more enriched at the end of the screen, for example, for a positive selection screen. The protocol provides for both a lipofectamine version and a PEI version for producing lentivirus. In special cases, for example, for non-dividing cells, AAV can also be used. So for the screening transduction, the first step before transducing for the screen is to determine the titer of the lentivirus. we provide for 2 different methods of transduction. Either the spinfection method, which will work for a lot of cell lines, or the mixing method, which is more often used for stem cells and primary cells that are more sensitive. And this is very important to determine the multiplicity of infection for the screening transduction. The multiplicity of infection is defined as the number of cells that are surviving selection normalized by the cells that do not have the selection. And typically for the library, we'll transduce the guide library at an MOI of less than 0.3. This reduces the chance of any cell receiving multiple guides because that will make the downstream analysis very difficult. And then after determining the titer of the screen, we will scale up the screen accordingly. So what do the numbers look like for scaling up? For a library of 100,000 guide RNAs, typically we recommend 500 cells per guide RNA for the coverage of the screen. This number can increase for more noisy screens, for example. But typically at the baseline, we recommend 500 cells per guide RNA. So if you are transducing at an MOI of 0.3, that means only 30% of the cells survive at the end of the screen. And so we need to start out with 167 million cells. So that's a lot of cells, and that's why in a lot of cases, we would recommend using a targeted screen instead of a genome-scale screen. And then during the screen, we will recommend maintaining this coverage. So every single passage maintain about 50 million cells for a guide RNA library with 100,000 guide RNAs. So again, a lot of cells. So just going through the knockout and activation separately, for the knockout libraries, there's an option to make the Cas9 cells first. And the Cas9 expressing cells can be made by transducing the Cas9 at an MOI of 0.7. And then after making the cell line, we transduce the library at less than 0.3. For the screen, typically for knockout, we recommend starting the screen 7 days after transducing the guide RNA library. This is because the indels saturate at about 1 week. And so you want to wait for the indels to saturate first before starting the screening selection process. And again, during the screen, maintain representation at greater than 500 cells per guide RNA.
Why the Cas9 MOI less than 0.7?
So this is, okay, so if you transduce at higher than that, you'll get a fairly wide distribution of Cas9 expression. That means that when you transduce the guides, each guide is going to have like a distribution of Cas9 expression, and that may affect your indel rates, right? So we want to also try to cap the Cas9 at less than 1 or 2 copies per cell, and that's why it's less than 0.7. But it's not as stringent as the guide RNA library. Yeah, so the Cas9 is stably expressed when we transduce it. So it's a lentivirus carrying a Cas9, and it gets integrated the genome, and then stably expressed in the cell line. Okay, so during the screen, after we make the cell line, when you're transducing the guide RNA library, remember to do 2 to 4 bioreps of the screen. The bioreps are very important for calling the hits because usually you'll try to call the hits from 1 biorep and then take the hits that are overlapping multiple bioreps. Again, because the screen is a very noisy process, so if a hit shows up in multiple different bioreps, the chances are it's real. If a hit only shows up in one of the 2 to 4 bioreps, then it's probably it probably was enriched as a result of noise. So similarly, for the transcriptional activation screen, we transduce the activator components at an MOI of less than 0.7, transduce the guide library at an MOI of less than 0.3, and then for the activation, because the activation acts much faster than the knockout screens, you can start the screen as early as 4 days after the library transduction. So for during the screen, there are many considerations, the most important of which is to choose screening parameters that maximizes the difference between your experimental and control conditions. For things like a drug selection, we normally pick the IC50 value of the drug for that cell line because you don't want to too stringently select the cells. That will lead to a lot of cell stress, and you also don't want the selection to be too weak. And so typically we pick the IC50 value. Again, during the screen, we still maintain the coverage of over 500 cells per guide. And then for the first time that you're setting up the screen, typically I would recommend collecting multiple time points throughout the screen because for dividing cells, you will always have extra cells, and it doesn't hurt to freeze down cells at 1 week, 2 weeks, 3 weeks after starting the screen. Just in case you want to go back and look at an earlier time point, you can just take your frozen cells and harvest the genomic DNA. And during the screen, it's very important to keep all the protocols very consistent. If you change anything, sometimes you can't tell if it's because you switched out like FBS in the middle of your screen, or if it's actually because of the screening selection. So the next step is to harvest and analyze the screen. And we also provide a large-scale genomic DNA harvest and PCR and PCRP process for this. And the NGS process is very similar to the library amplification for a QC. So for the screening analysis, most screening analysis algorithms follow a general theme where you normalize the experimental condition, the guide counts in your experimental condition to the guide counts in your control condition. Then you rank the guides based on the fold change. And then for each gene, you look at where the guide rankings are. And if there's multiple guides that are fairly enriched, then you call that gene as significant. In the Nature Protocols paper, we cover rigor because that's the analysis program that we've used for most of our published screens. But there are more recent analysis algorithms that might be more helpful, like MAGIC or STAR might be worth looking into for screening analysis. And then typically, we take the average or overlap of the top genes from each biorep to call the hits. All right, and the very last step of screening is the validation. As I mentioned, screening is a very noisy process, so you want to verify individually whether or not your guides actually lead to the phenotype that you want. So for validation, we go back and we package each guide RNA that was enriched in the top hits individually. And there's a cloning process for cloning single guides. We use Golden Gate assembly for this. We package this into small T25 worth of lentivirus, transduce this, and for knockout, we have a protocol for extracting the genomic DNA from the validation cell lines. Then we provide a 2-round PCR of amplification of the target site with the Illumina adapters. For this, we use the 2-round PCR instead of the 1-round because it's much more versatile and it's easier to combine the primers with the barcodes and the Illumina handles with primers that target different sites in the genome. And after you transduce the cells and verify that there are indels, you need to verify that the screening phenotype is actually there and that the target protein was actually knocked down because the target protein is not always knocked down. And so it's very good to verify that with a Western blot. For validation of transcriptional activation, we provide a homebrew protocol for rapid extraction of RNA and RT. This is a very similar process to the commercial cells to CT protocol, where you have a 96-well plate, you add lysis buffer, mix a couple of times, stop the lysis, and then go straight from that into the RT process. So it's very fast. It usually takes me about 7 to 10 minutes to do the entire process, and because it has no columns, it's much faster. And it's also much, much cheaper than the commercial cells to CT protocol. So after you do the RT, we recommend Taqman qPCR for amplifying the gene of interest because the Taqman qPCRs have 3 I guess, so there's 2 primers and there's an additional probe, but that adds a layer of specificity that something like Cyber doesn't have, for example. And after verifying the activation, again, similar to the knockout, you need to verify the screening phenotype and the Western blot to verify that your protein was actually upregulated. All right, so lastly, I'd like to go over, again, what the most important screening considerations are. During the screen, like I mentioned many times, we need to maintain coverage at greater than 500 cells per guide RNA throughout the screen. Another very important thing is to include all of the necessary controls. This includes the non-targeting guide RNAs in the library and sgRNAs that target genes that are known to affect your phenotype. These type of sgRNAs are, if you know what they are, if you know what the genes are that are affecting your phenotype, are very helpful in setting your screening parameters. And of course, you always want to include a condition without the screening selection. This is your control condition that you will normalize the experimental condition guide counts to. So it's also very important. And then finally, to remain consistent throughout the screen, you use the same protocols for screening and validation. And this will help get you the genes with the phenotype that you want. So lastly, I'd like to thank all the people who helped me put together this protocol, especially Silvana Kahneman, who was the early Cas9 activation pioneer. So thank you very much.
Thank you so much. Where can we find the script that you mentioned that would help us design?
Oh, the script, all the scripts that are required for screening are published in the Nature Protocols paper.
Even the one to design guide RNAs,
yes.
like custom? Okay.
Yes, the custom ones are also on the Nature Protocols. And I keep a more updated version on our lab's GitHub page.
So when I email out the slides, I can also email out a PDF of that protocol. And that protocol too, at the end will, it has the web address for the lab's GitHub. So you can just go to that link and get any of the updated scripts you need.
When you're doing an activation screen, is it possible that some genes you might miss because the position of the guide RNA is in inaccessible chromatin regions?
Yeah, so in the original SAM activation paper, they tested quite a few guides, and it seems like the region 200 base pair upstream of the transcriptional start site is the best region for targeting, and we use that guide design for the entire library. Now, for closed chromatin regions, from my experience using the activators, I've activated quite a few silent genes that are in closed chromatin regions and have had no problem with that. I think it's because the Cas9 is a fairly large protein and it can open up the chromatin. So.
Very nice talk. Do you have protocols for to attack the GC-rich regions?
Sorry?
Do you have protocols that will attack GC-rich regions?
Typically for the guide RNA design, we'll filter out the GC-rich regions because it's harder to synthesize the oligos. But that doesn't mean that you can't target the GC-rich regions. You can definitely still target the GC-rich regions. It might just be a little bit more difficult to clone the guide and to sequence the guide, but it's definitely doable. And it's the same protocol.
Okay, any other questions about screening? Are we ready to move on? Yeah, okay. All right, thank you, Julia. All right, so. Okay, so we'll do one more sort of about 20 or 25-minute presentation. This one will focus mostly on some of the newer Cas enzymes that target RNA rather than DNA. We will have two other graduate students from the Zhang Lab, Jonathan Gutenberg and Omar Abudea, who have done a lot of the work to discover and characterize and adapt these enzymes as tools. And then again, just a reminder, so after this is over, then we'll kind of break it down into small groups, and any other questions that you guys have, or if you want to talk about the specifics of your experiments, we'll have some more people from the Zhang Lab come on down, and we can help hopefully get all of your questions answered.
diverse, and Cas9 is actually just a really small slice. So, I think you spent the past couple hours learning a lot about what Cas9 can do, and it's really powerful. But there's actually a lot of other types of CRISPR enzymes, and they're pretty complex. They're made of many different types of subunits, and a lot of these have really not gone beyond bacterial cells because they're too complex to use in mammalian cells. But we, a couple years ago, had this question about whether we could find more systems like Cas9 that just have a single protein, they're much simpler, and could have new biotechnological applications. And to do this, we kind of looked at what proteins would be highly conserved across all CRISPR systems to kind of come up with new signatures for pulling out different systems. And so what's really interesting when you look at the different types of systems, there's type 1, 3, 4, and the type 2 systems which have Cas9, and type 5 which have CPF1, is they all, almost all of them contain Cas1 and Cas2, which are the proteins important for the memory of an infection. So remembering that you've been infected, and so that the system can then be immune in the future. And so what we can do is take these enzymes and actually use them to pull out areas in newly sequenced bacteria or bacterial genomes online of potential places where there might be a CRISPR system in the bacteria. And when we do that, we can actually come up with new classes of CRISPR systems. So we did this a couple years ago as a collaboration with Eugene Koonin at NIH, and we had many hits, and we spent time trying to understand what they are and how they might be different. So CPF1 was one of these hits that came up first, and we eventually found it to be a DNA targeting enzyme. So it was kind of like Cas9, but it had a simpler guide and had different features that kind of make it amenable for slightly different applications than Cas9. There was C2C3 and C2C1, which are like CPF1, and then there was this other family, C2C2, which we found to be very interesting because it actually did not have any DNA domains. So when you look at the alignment of an entire family, you see what's highly conserved are these two FN domains shown in dark purple, which are known to be RNase domains. And because they had these two RNase domains, we thought maybe this could be a programmable RNase. And so we did a lot of sort of functional genomics in bacteria, biochemistry work, to eventually characterize C2C2, which is now named Cas13, as an RNA-guided RNase. And the basic idea is it's fairly similar to Cas9. You have an enzyme, Cas13, that's expressed, and you have a CRISPR array that's also expressed. These CRISPR arrays can be chopped up by, actually now we know it's Cas13, chops them up into mature CRISPR RNAs. And these CRISPR RNAs guide the enzyme to find sites in RNA targets to bind to, much like the Cas9 enzyme, and then to cleave. And this is where it differs. Instead of cleaving in a defined site, it'll actually generate many cleavage sites anywhere in the RNA target. So it basically degrades the RNA target in single-stranded regions. And so we found this to be pretty interesting because we envisioned kind of a lot of applications for RNA targeting and how an easier tool to do this could be very useful. But there was one problem, and it's this activity, when it became activated, it actually didn't just cleave in the target, but it would cleave any other RNAs that were nearby. So what we saw is if you had a CRISPR RNA and an RNA that's not complementary with Cas13, you get no cleavage of this non-complementary RNA. But if you had a CRISPR RNA that bound to its target and activated Cas13, and you had this non-complementary RNA, you would also see cleavage of this non-complementary RNA. And we call this the collateral effect because RNAs nearby are collateral of this target recognition. And so this was kind of disappointed us at first, but we realized we could actually use this as a detection technology because, how's that? Well, yeah. Okay, so we realized that we could probably use this as a detection technology because the recognition of this target results in this part of the talk where we will talk about how we can use Cas13 for detecting nucleic acids. So the idea is if you have a complex pool of nucleic acids and there's this blue target that you want to recognize, you can have a CRISPR RNA that recognizes the blue target with Cas13. And what it'll do is activate cleavage of the target. Now, if you have another RNA solution as a fluorophore quencher, this probe can also get fluorescence. And so you can use this as kind of a cleavage reporter for the presence of your targets. And so while this worked pretty well, it was only getting us down to femtomolar level sensitivity. And so femtomolar is kind of like 100,000 molecules per microliter. And we really wanted to get down to a single molecule per microliter. So to do that, as part of a collaboration with Jim Collins' group, who's done a lot of work in nucleic acid sensing, we combined this part of the detection with an amplification step. And so recombinase polymerase amplification can take DNA or RNA and amplify a lot of DNA. It's kind of like a PCR, but it works at 37 degrees. So it operates at a constant temperature, and in 10 to 20 minutes, you can amplify a lot of DNA. And as part of the primers, we added T7 promoter, so you can convert that DNA that you amplified to RNA, which then can be read out using the Cas13 detection scheme. And all of this actually works in a single reaction, so it's all compatible, and you can kind of make basically one master mix that takes DNA or RNA in, and it gives you signal out on the other end. And so basically all you need to do is design 2 primers around an RNA target for the RPA, and then in the middle, kind of like a Tacman probe, you design a CRISPR RNA that has complementary through the spacer sequence, kind of like Cas9. And so all you need are basically just 2 simple primers and then this guide RNA, which is actually really cheap and easy to make, and you can design kind of a Sherlock assay. So it's a bit simpler than Tacman, a lot cheaper, and the probes are a lot quicker to order because you don't have to have a fluorophore quencher on every new sequence probe you design. So, and what we found with this approach is that with Cas13 without the RPA amplification step, you get this femtomolar sensitivity, which is right here. But with amplification, we actually could shift the curve and get sensitivity down to 2 attomolar, which is down here. And so 2 attomolar is about a single molecule of nucleic acid per microliter. And this works for RNA, which is here, and also DNA, which we get down to 2 attomolar, even stochastically below 2 attomolar. So by just sampling less than a molecule per microliter. So, so we validated the concept, and we wanted to show it in a few different applications. So one that we thought would be really interesting is Zika diagnostics. The current Zika diagnostic space is not very good, and so it would be useful to have a better one. So we collaborated with Pardis Sadeghi's group here to get all these samples she had from patients. She has serum and urine from patients that have been infected with Zika. And we can actually input them into the Sherlock workflow. And what we find is we can actually get down to single Zika genome copies per microliter from actual patient samples. Now, this was, we RT'd it and RNA-seeded it ourselves before we put it into Sherlock, but we've done follow-up work where you can actually just directly heat treat the sample and put it into the Sherlock assay and get actual detection in less than an hour without any nucleic acid extraction or any complex steps. So it's pretty exciting how far we can kind of push this approach. We've also been able to do this with detection of Zika reporter fragments and lentivirus particles with freeze-dried Sherlock components. So we can actually freeze-dry the components on paper and then just add the sample and reconstitute the components to get detection. And this also works down to 20 attomolar. We get a little bit of a hit of sensitivity, but we can still do down to basically 10 molecules per microliter with just freeze-dried components that can last for a long time and not require any kind of cold storage. So, so that's really nice. The last thing I want to talk about for Sherlock is it is really specific. So I kind of did the Tacman qPCR comparison earlier. It is actually able to detect a single mutant or SNP or mismatch. Basically because if you have a SNP within the guide region, you can actually engineer Cas13 to be very sensitive to a mismatch and only get signal if the mismatch is present. And so we designed an assay against the mutation involved in BRAF V600E, which is a mutation in melanoma. And with a wild-type sensing CRISPR RNA, you basically get detection of the wild-type DNA in the blood sample, whether you have 100% wild-type or slightly less because you have some of the mutant allele now present. But for the mutant-sensing CRISPR RNA, we can actually get quantitative detection of different allele fractions of the mutation in the cell-free DNA samples. So that was kind of a nice result, and shows that because these enzymes are so specific, we can sense mismatches. So, and then just kind of a little aside for how we do that is, it's interesting, Cas13 is only sensitive to double mismatches or more. So if you have a single mismatch, it will cut. But what we can do is actually engineer an additional mismatch in the guide RNA. So what's shown here on the bottom is the target of the mutation right here, and the CRISPR RNA that's complementary to it. What we do is we engineer an additional mismatch in the guide RNA. So that when this is trying to sense this mutation, it only has a single mismatch, and we'll give that signal. But when it's against a different mutation, it will have two mismatches, and that won't give signal. So it's a little bit of engineering of the guide RNA to give you single mismatch specificity. And you can see then that you get a very nice signal for the Zika African strain versus the American strain, which is a single mismatch difference. So in general, Sherlock is a really nice detection platform for many different types of applications across infectious disease, point of care, and genotyping, as well as detecting cell-free DNA in blood samples. And the main key takeaways of the technology is it's very sensitive down to single molecule per microliter. It's very specific, it sense mismatches, and it's also really cheap. The components we calculate, from the cost that we buy from the vendor are just 60 cents per test. And it works even better in PCR because you can use crude samples as the input. And we're kind of working on non-fluorescent readouts now to make it even more portable, so like colorimetric or kind of like pregnancy strip readouts based on lateral flow designs. And it's really quick, so a lot of the assays give us less than an hour readout, and some of our new work is trying to get it to less than even 20 minutes or so. So it's still a work in progress, but kind of a powerful tool for detection. So now I'll hand it over to Jonathan.
Cool. Thanks so much. All right. So Omar gave a great introduction and one application, which is single molecule detection. And of course, I'll be talking about using Cas13 enzymes inside of mammalian cells to actually target RNA for degradation and other things. So you probably spent more time than you can handle about Cas9, but it is pretty much an amazing enzyme. So we can do a lot of things with it. We can cut DNA using its natural endonuclease domains, but beyond that, we can deactivate those for transcriptional control. Is that better? For transcriptional control. So we can, Julia talked about the SAM system for upregulating genes. We can also downregulate them. Going even farther beyond that, we can actually put epigenetic modulators, so we can actually change histone modifications or DNA modifications to see the cause and effects of those. And of course, we can actually label DNA and see what it's doing. So there's this whole slew of things that we can do, which really begs the question, what can we do with a tool that can bind to and or cut RNA? So today I'll be talking about two things that you can do. One is binding to and cutting transcripts for knockdown, and then a little bit of work that we've been doing on catalytically activating the protein and actually having it specifically bind to transcripts for imaging and localization in live cells. So we knew that Cas13 worked inside of a test tube and inside of bacteria, but the first question was, can we actually get it to work inside of mammalian cells? So we designed a reporter vector that has two orthogonal luciferase, the luciferase from Gaucher and the luciferase from Cryptidina, or GLUC and CLUC. And we can express both these luciferase transcripts along with our Cas13A and a targeting CRISPR RNA or guide, and then we can target one of them. We like to target GLUC, and then measure the luciferase ratio as an indicator of our targeting efficiency. And we wanted to try this out with a couple different functionalizations of the Cas13. So one great thing is that since Cas13A is a totally exogenous protein when we're introducing it, we can put things onto it, like nuclear localization sequences or nuclear export sequences that allow it to be cytoplasmic. So we decided to localize into two places, the cytoplasm and the nucleus. And we also wanted to compare it to an RNAi kind of regular standard with a small hairpin RNA. And we found that for a couple different guides, we chose 2 guides to test, we could get essentially equivalent or better knockdown compared to shRNA. We also found that our nuclear construct was the best at targeting. We don't have exact reason why this is potentially more stable in the nucleus or it's easier to target an RNA, but one advantage, as you'll see later, is that we can actually target an RNA in the nucleus. So given that we can target a single reporter transcript, we decided to go kind of far beyond that. And we wanted to see if we could target an endogenous transcript. We didn't really know if we would be successful with our first guide that we tested or another guide that we tested. So we decided to go for a larger, unbiased approach. We actually designed a tiling library across the entire transcript. And we could then do plasmid cloning. So this wasn't a pooled screen, as Julia discussed. We don't yet have lentivirus working for Cas13a, but instead we decided to do it in individual wells. But we can use the same Golden Gate cloning to actually clone pretty easily about 96 different constructs. And then we can actually transfect these with Cas13a and measure expression of our targets by qPCR. So we first targeted a cytoplasmic messenger RNA, PPIB, and we found that there are actually fairly significant regions where we can get fairly good knockdown, about 50% or greater for different guides. And we hypothesized that the variability among the transcript is partially due to transcript accessibility. Cas13 doesn't really like to really bind to or cut double-stranded regions, and we do find relationships between this targeting and predicted in silico folding. But it's also possible that it's just due to guide sequence. So we could target a cytoplasmic gene. Then we wanted to see if we could target a nuclear gene. So we chose mallet1, which is a nuclear long non-coding RNA. And we can see that we have guides that about reach 50% knockdown, which is okay, but except when you think about comparing that to RNA interference. So RNA interference relies on cytoplasmic machinery and is not very good at targeting a nuclear transcript. And when you compare our Cas13a knockdown of mallet1 to the position-matched guides with shRNA, you can see that shRNA just is not capable of knocking down mallet1, or we can achieve about 50% or more knockdown when we select these guides. So we see Cas13a as, because it's exogenous, having this flexibility where you can actually target transcripts in the nucleus or in the cytoplasm exclusively. Another great advantage of Cas13a is that, as Omar mentioned, it actually processes its own pre-CRISPR RNA into a CRISPR RNA when it's expressed inside of bacterial cells. So if you take something that the Cas13a thinks is a pre-CRISPR RNA, it'll actually chop this up into individual guides that can then be loaded and used for targeting. So this provides a free way to multiplex under a single promoter. And we decided to target 5 different genes. So PPIB, CXCR4, and KRAS are going to be essentially cytoplasmically localized, and TINKER and PCAT are long encoding RNAs. And we wanted to compare essentially our multiplexing and our single guide approaches. And we found that for almost every gene, TINKER we couldn't get exactly, we can get great knockdown with our single multiplexed array that's comparable to a single guide delivery. So if you would like to target 5 genes or potentially even like 10 genes at once, Cas13a allows you to do that under a single promoter in a very condensed way. So this whole construct is about 250 nucleotides long. So it's very small. We also wanted to think about the specificity of targeting. So specificity when you're doing screening work or even direct targeting work is very important for having a robust experiment. And because Sherlock is so great at specifically targeting transcripts in vitro, we thought that that might extend to in vivo with Cas13a. So we took our successful luciferase guide and we introduced single mismatches or double mismatches. And we found that for single mismatches, we can get a mild reduction in targeting efficiency, especially when they occur near the middle of the transcript. But if you can engineer 2 double mismatches, then you can actually get pretty drastic reduction in activity. And so a similar approach, as Omar alluded to in the Sherlock section, where if you want to sense a single mismatch, you can introduce your own additional synthetic mismatch, which doesn't hurt you much unless you have your mismatch of interest as well, could allow for single mismatch targeting as well. And so this provides an effective way to actually target a SNP or a deleterious allele, for example, in a live cell. We also thought that this selectivity might have an effect on the actual off-target effects genome-wide or transcriptome-wide. And it's known that RNAi has problems sometimes with specificity, which is why a lot of people turn to CRISPR for screening. So if you actually want to quantify or see the specificity of RNAi, you can do a non-targeting shRNA and a targeting shRNA and see how they correlate together. So we did that, and this is essentially counts for transcriptome-wide treatment. And you can see that there's this fuzziness across the axis. And this corresponds to genes that are either upregulated or downregulated due to off-target effects. Ideally, we'd want it to be essentially a straight line. So if we repeat this experiment with Cas13, you can see that it's much, much tighter. We don't see these off-targets lying off the axis. We do see this reduction of this red point, and that's our targeted luciferase, but there's not much else there. So our reviewers demanded that we do some math. No. If you'd like to quantify this, you can actually do differential gene expression. And you can see that if we count differentially regulated genes, here they're 2.5-fold upregulated or 75.75 downregulated, and we look for significantly changed genes. You get about 1,000 off targets almost with certain targeting RNAis, and we don't have any. So we find this as a pretty compelling advantage for, when you like to do knockdown. I mean, it's essentially able to have no detectable off targets. So this is usually the point of the presentation that people ask, well, there was a whole in vitro part about actually this effect where there's promiscuous cleavage due to targeting. Why don't you see anything? And we essentially do not know why. We don't have a great ironclad hypothesis for why the collateral effect does not occur in mammalian cells. It's possible that there's small amounts of cleavage happening that we can't detect or that we can't access ribosomes. But we wanted another way to show that we weren't having any sort of growth phenotype. So we actually looked at how the cells grew. So when we target luciferase, we can see a very effective knockdown with different guides, either from a screen or from another experiment. So it's definitely targeting luciferase. But if we look for growth changes, we can actually see that when we assay cell growth by cell titer glow, we don't see any sorts of significant growth changes in our targeted cells versus our non-targeted cells. And they're also not losing the Cas13a plasmid because we can either select or non-selected. And in both these cases, the Cas13a is tagged with a GFP. And you can see that the cells don't actually lose any sort of fluorescence either. So we know that it's not having any sort of growth effect on the cells, and we don't have any visible off-targets. So we conclusively say that the collateral effect does not have any sort of detectable phenotypic changes in cells when you target it. Although it's possible in other cell types we may see something different. So given that we can get very good specificity, we wanted to think about how we could actually use Cas13a for another application, and that was transcript targeting. So to test if Cas13a was even targeting and binding to transcripts, we first catalytically inactivated by mutating its conserved HepN domains. That's dCas13a. And then we wanted to see if we could target our luciferase and pull it down. So it's our Cas13a is HA tagged, and we can actually immunoprecipitate it and check for overexpression or enrichment of luciferase by qPCR. And you can see that if we target a GLUC mRNA, we can have significant enrichment with 2 guides by qPCR over our non-targeting condition. And this also holds for an mRNA targeting actin. So we were encouraged that, one, it's binding to RNA, and we could take this further. Also, this could be used to purify RNAs of interest and detect other things on them in a co-IP sense. But we want to take this farther and think, could we actually use this as a platform for visualizing inside of a mammalian cell? So there's one slight nuance with targeting a fluorescent protein inside of a mammalian cell, and that's we can't determine whether it's bound or unbound. So binding is very good if this were a very focal phenotype. Maybe we could distinguish that. But we need to find a way that we could actually dynamically regulate the expression and fluorescence of the whole cell based on targeting. And we actually made what we call a negative feedback, or dCas13a NF. So the way that this works is that we have our Cas13a, and it's got a fluorophore on it, a superfolder GFP. We also have a zinc finger and a crab domain. And that crab domain is a repressive transcriptional domain, and the zinc finger actually targets back to a zinc finger binding site on the promoter. So what happens is that when Cas13a is nuclearly localized, it will bind to and repress its own transcription. However, if Cas13a binds to a transcript either right after it's translated in the cytoplasm or as that transcript is exported, it'll be taken to the cytoplasm. And then it cannot suppress its own transcription, and more will be expressed. So it's a negative feedback loop that will actually tune the amounts of the imaging construct so we can target things that aren't just really very focal phenotypes. So what you can see is that when you have a non-targeting guide, it's essentially nuclearly localized and very dim. But when we target actin, for example, we can see pretty impressive translocation to the cytoplasm. So you can see essentially these are cytoplasmic and much higher expression than in our non-targeting case. And we wanted to actually use this to detect the occurrence of a phenotype. And these are fixed cells, but we also do this in live cells. And so we wanted to look at the accumulation of actin in stress granules under a chemical stress. So what we did is we used G3BP1, which is a great marker for stress granules, and we targeted actin and treated them with chemical stress. And you can see that we can get very good overlap with our targeting construct and the stress granules, but our non-targeting construct essentially stays in the nucleus and doesn't even come out to label any RNAs. So we see this as a great way to actually image phenotypes in cells. It works the best with a focal phenotype, but we're working on making it better for actually imaging transcripts or any other RNAs at hopefully a single molecule resolution. So this is, we see as kind of the start of a toolbox of RNA tools. So with DNA binding CRISPR tools, you can do a lot of things. You can cut, you can image, you can change expression. And now hopefully we're starting to add to the toolbox. So these represent cutting, but there's a wide range of applications. So by fusing different domains, you can modulate translation, modulate splicing, assess localization, which we've done, but also alter localization. And you can modulate, of course, RNA levels or change their function or even edit them. So we see a lot of future potential for the tool in mammalian cells, and we're really interested in people using it for many different things. So on that note, Omar and I would like to thank the entire Zhang Lab and, of course, Feng. I'm also co-advised by Aviv Regev, and specific members of the Zhang Lab who helped a lot. Patrick Eslet-Bickler was a great visiting master's student who helped a lot with the Sherlock and the mammalian stories, and Brian Franklin and Max Kellner are current lab members who have been doing a lot of amazing work on mammalian and Sherlock. So, and of course, Feng and our collaborators. So, thank you.